In this video, we're going to run through one of the required practicals for upper sixth. So this is an investigation into measuring the rate of respiration in yeast. So we're going to go through some of the background information and the key fundamental science that we need to understand for this practical, and then also consider things like the methodology, any safety aspects. And hopefully from this, and we will also watch a demonstration of the practical being carried out, you will not only consolidate your understanding of respiration, but you will also be more prepared to answer exam style questions if a question on this sort of investigation were posed to you in an exam setting. So let's talk first of all about yeast. This is the organism that we are going to use in this investigation. So yeast is a single-celled fungus. Okay, so it's eukaryotic cells, and we are familiar with yeast. We have it used in all sorts of food production. So what do we need to know? We need to know that it can respire both aerobically, so using oxygen, and anaerobically. It's this anaerobic respiration or fermentation that is used in things like brewing and the production of alcohol-based drinks, because yeast, when it respires anaerobically, produces ethanol. That's why it's used by these industries. We're also familiar with the idea of yeast being used in baking, because if you are to put yeast into your dough and it respires, it produces carbon dioxide, which causes bubbles to get trapped in the dough and make the bread rise. But today we are going to focus on just aerobic respiration, and there is a reason for this. So during aerobic respiration, we now hopefully all understand that this means that all 4 stages of respiration are allowed to continue. So we have glycolysis, and then we are able to move on to the link reaction, Krebs cycle, and then finally to the electron transport chain, which involves oxidative phosphorylation. Now this is the important bit. It's this movement of electrons that we are going to take advantage of in this investigation. So in this investigation, the movement of electrons is going to enable us to hopefully visually gauge the rate of respiration. How are we going to do this? Well, we're going to use a substance called methylene blue. Now, methylene blue is very interesting. It is, and you will see this in the demonstration video, but it is a very blue substance. Now, when it gains electrons, so when it becomes reduced, it actually becomes colourless. So the logic is, if the yeast that it's mixed with are respiring, then there will be electron movement. Now this means that the methylene blue will be able to accept these electrons, or at least some of them, and start to become colourless. And from this we can gauge the rate of respiration, because obviously if respiration is happening at a faster rate or a greater rate, we would expect the time taken for the methylene blue to become colourless to be much quicker. So a shorter time to go from blue to colourless. So this is how we are going to measure the rate of respiration in this investigation. So we will measure the time taken for the methylene blue to become colourless. So we're going to take yeast, we're going to ensure that it has access to oxygen, so it's doing aerobic respiration, and we're also going to give it a respiratory substrate. So we're going to give it glucose so that it can actually carry out respiration effectively. What we will vary though, so this is going to be our independent variable, is we're going to vary the temperature. We're going to use 2 different temperatures. One will be around about room temperature, and the other will be between 35 and 37 degrees Celsius, so just slightly warmer, closer to body temperature. What we are hoping to see is that as we increase from room temperature to 37 degrees, obviously our yeast should not be adversely affected by this. The enzymes present within yeast shouldn't be denatured by 37 degrees. However, that extra thermal energy should be converted to kinetic energy, and hopefully what that will result in is more frequent successful collisions between key enzymes involved in respiration, and therefore more enzyme substrate complexes and a greater rate of reaction. Now, obviously, when we have an experimental hypothesis, we should have a null hypothesis. That means that if we were to carry out a statistical test to gauge significance, we will then be able to either accept or reject the null. So in this investigation, our null would be that temperature will have no effect on the rate of respiration in yeast cells. So as we've said, we're going to collect some data for 2 different temperatures. We're going to take some repeats. In this demonstration, we're going to take 3 repeats. Remember, the value of taking repeats is that we get more reliable results, we are able to calculate means, and we can discount or identify and discount any anomalies. Now, we do need to consider safety, and that's a key one of the CPACs. So we have all of the usual safety considerations to make of being in a lab. So many of the points that we know about, such as trip hazards, not running, not eating, not drinking, etc. But specifically for this practical, we are using methylene blue, which you are probably unfamiliar with. So what do we need to know about methylene blue? Well, it shouldn't be ingested. You mustn't eat or drink it. And it has vapours that, if you inhale them, can be also quite dangerous. It's also an irritant to things like eyes and skin. So what are we going to do to make sure that we are not at risk? So we're going to use an incredibly diluted solution. You'll hopefully see on the video just how low the concentration is. And then also general sensible behaviour. So we're going to have PPE, so we're going to wear goggles, eye protection. We're going to make sure that we clean up any spillages that may occur. And we're absolutely going to make sure that we're not eating and drinking in the vicinity, because we don't want to accidentally ingest it. Okay, so hopefully now you can sit back and enjoy watching the demonstration of this practical in a series of short videos. Okay, so what we're going to do now is have a little run through a practical, a required practical, which is the effect of temperature on the rate of respiration in yeast. So, when yeast is carrying out respiration, as we hopefully already know, there is a lot of electron movement. We've talked about oxidation being loss of electrons and reduction being gain of electrons. Now, this movement of electrons, these redox reactions, we can use them to try and measure the rate of respiration. And the way that we do this is we use this, which is called methylene blue. Now, methylene blue, as you can tell by its name and also by looking at the video, is a blue substance. Now, what's important about methylene blue is that when it gains electrons, okay, so when it becomes reduced, it actually becomes colourless. So we are going to be able to see how quickly this becomes colourless, and that's going to tell us or indicate to us the rate of respiration of yeast. So we have got methylene blue. We have got a solution of glucose, so we've got a respiratory substrate there, with yeast, okay? That's been made into a solution, as I've said, with some water, and it's been kept at good optimum temperature in an incubator. So it's been kept at around about 37 degrees Celsius. So it's nice and active, okay? We have also got an electronic water bath set to around about 37 degrees Celsius. That's going to be one of the temperatures we're going to investigate. And then for the other temperature, we're going to investigate room temperature. So at the moment, it has just snowed. It is roughly 23 degrees in the lab. Okay, so that's going to be our other temperature. So I'm going to run you through the method. Follow along with me, and we will hopefully pick up upon some key points in this. So we have 6 tubes. We are going to do 2 temperatures, room temperature and the 37 degrees. And we are going to repeat 3 times. So each temperature repeated 3 times. So using a marker, I'm going to track my tubes. So this is going to be 23 degrees C. Repeat number 1. 23 degrees C, repeat number 2, and 23 degrees C, repeat number 3. Okay, So we will run the first one. So what we're going to do is make sure that this is well mixed so that we've got our yeast evenly distributed throughout the solution, so we don't accidentally take a sample with too few yeast or too many yeast, to keep it nice and fair and controlled. Okay, and we are going to add 2 centimeters cubed of this yeast mixture to our tubes. Okay, so we are going to use a syringe for this, okay, because it's the most appropriate equipment we have. And as I say, we've stirred it, so we just need to measure out our 2. Okay, and place that into our 3. Okay, and remember, we're not going to cross-contaminate any of our syringes in this practical. So fresh ones for each different substance that we use. Okay, now I'm going to pop this back in the incubator so that it doesn't cool down too much for our next round. So next thing we are going to do is we are going to place these tubes into their own water bath. Now we're using room temperature, so we can simply fill a beaker with some room temperature water. Okay, and then we can place the mixture into this. This is to leave it for, so I'll stop my timer, we're going to leave it for 5 minutes. Okay, so that this gets up to the correct temperature before we start the experiment. Okay, so we will now wait for 5 minutes. Okay, so we have reached our 5 minutes. Using a thermometer, we can just double check the temperature of the room temperature water. As I say, it's about 23, if you can read that. So these should hopefully have reached the right temperature. So now we can move on to actually adding the methylene blue to it. So, things to remember about methylene blue. You should be wearing eye protection for this, okay? If you look up on the HAS cards about methylene blue, it's not something that you particularly want to get into your eyes. Okay, so what we are now going to do is we are going to fresh syringe for the methylene blue. We are going to measure out 2 centimeters cubed of methylene blue, and we are going to add these to our tubes with our yeast and our glucose mix in there. Okay, so we'll do it one at a time because I don't have the speed to do it all at once, and then we will record the time taken. So make sure that your timer is reset. Okay, we're going to measure out our 2. Okay, make sure we haven't got any and bobs. Okay, and then we are going to add it in to tube number 1. So once we add it, we are going to shake it for around about 10 seconds just to make sure that it's thoroughly mixed, okay, and also start our timer. So mix it in, thumb over, start the timer. Okay, and then we are going to watch to see when we feel that the colour, the blue, has disappeared. Welcome back. So in that pause, I have repeated this for all 3 tubes, and hopefully you can see that the result is that it has indeed gone colourless. So the time taken, roughly we had 220 seconds for tube 1, 236 for tube 2, and 219 for tube 3. So we'll be able to work out a nice average for that. Okay, so now we are going to move on to our second round of experiments. Okay, so we are going to be investigating the other temperature now. So for this, we're going to be using the electronic water bath, which is set to a higher temperature. So as I say, at the moment, last time I checked it, but it's always good to double check using the thermometer, it went up to roughly 37 degrees Celsius. Okay, benefit of an electronic water bath is that obviously they are thermostatically controlled, so you get a far greater level of control than, say, if you were to use hot and cold water or a Bunsen burner. Okay, so we know that that is set to a good temperature. We'll leave this in the cradle because we'll put the entire cradle in there in a second. So same thing again, we're going to take 2 centimetres cubed of the yeast and glucose solution. We're going to place it into these tubes. We are going to let them reach temperature for 5 minutes, and then we are going to add the methylene blue and see how long it takes for it to become colourless. Okay, so hopefully you have all predicted that at this higher temperature we should get a faster rate of respiration, because it's not so high that it should cause any denaturing of key enzymes, but it is high enough to increase the kinetic energy and hopefully increase the rate of reaction. So you can see the cradle is there. We will put the whole of this in the water bath. Let me get the yeast. Same system again. We're going to use a dedicated syringe for this. We're going to make sure that we have thoroughly mixed in case it has settled again, so that we get evenly distributed yeast and hopefully even samples. And we are going to measure out our 2 centimetres cubed. Apologies about this alarm that's going off outside. I did try to wait, but nobody seems to be turning it off. Okay, yep, that's 2. And check that. Yep. Okay, and again we're going to carefully pop this into the water bath with our thermometer, and we're going to leave that for 5 minutes. So our 5 minutes have passed. This should be up to temperature. So we move on with the final step, which is again our 2 centimetres cubed of the methylene blue being added, a shake for 10 seconds, and then we will return our tube to the water bath. And we can put the other 2 in there as well, they're just going to stay at the same temperature. So tube number 1, in goes our 2. Give it a shake and start our timer. Seconds. Okay, and then we'll pop it all back in the water bath and keep an eye on it becoming colourless. So position it so that you can see, hopefully as well as possible, and don't shake it anymore. Okay, so again I have managed to, in the intervening time, repeat this for all the tubes, and you can see hopefully that they have all become colourless, and this was far quicker. So what we had this time is we had roughly 2 minutes for each. So tube number 1, that changed colour in 126 seconds. Tube number 2, that took 112, and tube number 3 took 97, which is slightly lower, but we can take an average with that now. So the last thing to do is just clean it all up, and then we can take our data and we can analyse it.